I will start sharing my screen. Okay, able to see my slide? Right. Okay, good morning, dear colleagues and audiences who are here today to listen to the Dengue Symposium talk. And my topic today will be on dengue detection, advances and challenges. Right, so I will bring you through these contents today in my talk, which will cover a brief introduction on dengue virus and its properties. And we will also look into the past and present situation of dengue in Malaysia. And of course, the highlight of my presentation today is on the advances in diagnostic tools. So from conventional to point of care, what we have now, and brief also a brief overview on the challenges and future direction. Right. So here we have a brief introduction on the dengue genome structure. The structure of dengue virus was first discovered in 2002 and elaborated via cryo-electron microscopy technique. So they found 11,000 or 11 kb plus sense RNA genome and codes for 3 structural proteins, namely the envelope, membrane, and capsid, and additionally 7 non-structural proteins, which are the NS1, NS2A, 2B, 3, 4A, 4B, and NS5. And they are coded in this direction. From the 5' to 3', we have the C, the core, the pre-membrane and membrane, envelope, and all the non-structural proteins. So of these 10 proteins, the E protein is a frequent target to study the variants. Why? Because it has high heterogeneity, meaning that the nucleotide sequence of the E protein is highly differs from serotype to serotype. So it is a good target to study variants in dengue virus. On the other hand, the NS2B and NS3 proteases from all dengue serotypes share structure and function similarity. So these two are considered as the most powerful therapeutic target for development of effective drugs. Well, as some of you might have known, upon infection, NS1 protein is secreted in the host, and that can be detected using a rapid antigen test kit. So it's a good biomarker. Until now, to date, we are still relying on this NS1 protein to indicate acute dengue infection. Right. So a unique feature of dengue infection is the serotype shift phenomenon. A serotype shift is actually a phenomenon where one predominant serotype of dengue is being replaced by another serotype at any given time, any years, or any season. So the actual cause of this phenomenon is unknown. Why it's happening, it's quite not clear yet. However, it's interesting to see a pattern, although it's not consistent. For instance, if you look at the statistics shown here, the graph which I retrieved from this publication by Mudin, MOH 2015, we can see to a great extent that the serotype shift is being displayed here from the year 1990 until 2014. Initially, there was a predominance of dengue virus 2, and then dengue 2 was then replaced by dengue 3, and then dengue 1, and so on and so forth. But dengue 4, usually it remains at the background level. It's not so commonly found in Malaysia. So yeah, that's about the serotype shift. Right. So what is it like in dengue distribution worldwide in the recent 3 years? So from 2019 to 2020, there was a massive global outbreak of dengue, where cases spiked up to few hundred thousands annually in most of the endemic region of the world. However, in 2021, there seemed to be a decline in the number of cases, as you can view here in the graph, in the map shown here in 2021. Right. How is it like in Malaysia? What's the current situation? So we are moving into 120 years of endemic journey of dengue in Malaysia. And so here's a bit of a timeline of dengue outbreaks in Malaysia. Dengue was first discovered in 1902 in Penang. It was discovered in 1901, but it was only documented in 1902. And then after that, we had the first severe dengue outbreak in the country, which occurred in 1962, also in Penang. And after that, only in 1973, MOH made dengue as a notifiable disease due to following the outbreak in Xinjiang, which actually killed about 54 people, and dengue 3 was incriminated as the major serotype during that outbreak. Right, and then in 1997, we also had major outbreaks. The causative serotype was dengue 1, dengue 2. After that, we had many subsequent outbreaks, including in 2004, 2005, and then 2007, 2008, and 2015 to 17. All these are caused by, we saw a predominance of dengue serotype 1. Okay, so this statistic shows the number of dengue cases from 2000 to 2021, which I obtained from the iDengue website. So for those who are not, who have not browsed this website, so iDengue provides dengue statistics in Malaysia, including the hotspots, information on the hotspots, and so on. So here, as we can see, Malaysia had an unprecedented dengue outbreak from 2014 to 2016, where cases hiked as much up to 120,000 cases per year. And after that, we saw a slight reduction, and it picked up again in 2019, and again there was a drop in 2020 till 2021. So here also to outline the drastic decline in the number of cases in 2020, where the number of dengue deaths reduces as much as 20.3%. And one of the causes that could have contributed to this, why dengue has reduced so much in 2020, is of course because we were having another issue, which is the COVID-19 pandemic. So this paper captured some interesting findings where they noticed that during the implementation of MCO, which is supposed to break the COVID-19 infection chain, but it also had some positive effect on the onset of dengue cases. Where the highest reduction of new dengue cases were reported in the central peninsular region, followed by the northern peninsula and eastern coastal region. However, this situation did not remain long, where new cases of dengue started to increase during the MCO phase 3, obviously because during the MCO phase 1, a lot of restrictions were imposed where people tend to stay indoor rather than outdoor, so perhaps the cases have reduced. But during the MCO period, phase 3, a lot of leniency has been given, so people were still moving in and out, so that could have contributed to the increase in the cases again. Okay, so moving on to the laboratory diagnosis of dengue. So this is an overview of the current available laboratory diagnosis. We have virus isolation, which is considered as a gold standard. And here I have shown the uninfected and infected cell lines. There is a wide range of cell lines that can be used to isolate the virus. So this is an uninfected cell, and then this is the infected cell. So the virus isolation, the success of virus isolation is judged firstly based on the CPE, which is the cytopathic effect. Cytopathic effect is actually the morphological changes that is induced due to the viral infection or effect of the virus to the host. And then, so since this is very subjective, and it's usually explained or interpreted in the manner of scores, you know, I can say that this cell, the CPE is 1 plus, 2 plus, 3 plus, and so on. So it's very subjective. Eventually, we have to proceed to other assays to quantify the degree of the virus growth in the cell. For instance, we can use a plug assay, a focus forming assay as shown here, to check the viability of the propagated virus, or we can also proceed to immunofluorescent assay to capture the antigen. In addition to that, we also have antigen detection assays. So basically, it captures the antigen of the dengue Mainly, it's the NS1. So now, with advances in technology, we have combo kits, okay, that combines the NS1 as well as the IgM-Ig. So it's the antigen plus the antibody detection. So it's a very good POCT that is useful in the hospital settings, especially. Okay, then we have PCR. So real-time RT-PCR is a very useful technique that can be used to detect dengue and all 4 serotypes of the dengue. It has faster TAT than the virus isolation. So in IMAR, we use this technique, a real-time RT-PCR for detection and serotyping of dengue samples that are being sent to us. So the interpretation here is qualitative. It's either positive or negative, detected or not detected with the information of the serotype, whether it's dengue 1, dengue 2, dengue 3, or dengue 4. And then we also have laboratory diagnosis based on antibody detection. This is particularly useful in differentiating the recent and past infection. Well, recent infection is characterized by a high titer of IgM, whereas past infection is or secondary infection is characterized by the IgG detection. So what's new in dengue detection technology? So here, first, let's look at the virus isolation. As I have mentioned before, the virus isolation is a gold standard detection. Why? Because of its consistency, its capacity to generate more infectious viruses for future research, and excellent compatibility with a wide range of samples. You can isolate the virus using serum, blood sample, urine, and a lot other range of samples. And also, like for respiratory viruses, you can use the nasal swab and so. So we have a wide range of cell lines as well that is compatible or that can be utilized for dengue virus isolation, including the Vero cell and C63 cells. So basically how it is performed, the cell lines are inoculated with patient samples, example serum, and after which you observe for CPE from day 1 to day 7 usually. So after which the culture supernatant are harvested for subsequent detection or quantification such as plug assay, focus forming assay, or immunofluorescence. But there is a challenge with this technique. It is time consuming, obviously, because you need to wait up till a week to observe the CPE, after which you can only confirm the viability using some other assays. So it's very time consuming, it's laborious, and which is why there is a need to look into new advances in this area. So one of the new advances that I picked from this particular publication is the usage of biosensor technology to capture live viruses. So WASIC 2017 explained the development of a biosensor using single wall carbon nanotubes called the SWNT that mimics the in vitro cell culture system. So basically what the scientists did, they took the single wall carbon nanotube and they functionalized it with heparin as a biorecognition molecule that detects the virus. Now, dengue binds to a wide range of receptors, host receptors, in order for it to be internalized and replicate in the host. So one of the receptors is the heparin sulfate. So here the researchers used heparin, which is a close analog to the heparin sulfate, as a receptor to the 4 serotype. So they attached the heparin or overlaid the heparin to the SWNT, and then they went through a series of chemical treatment, and then this system was able to capture viable viruses via binding signaling. The more virus binds to the heparin overlaid in SWNT, the more signal it releases, and with that, they are able to come out with a quantitative method for detecting viable dengue viruses in patient sample. But what is the problem with this system? It is not able to support growth of virus. Obviously, you can't propagate the virus for future use. You can only detect it, so it stops till there. So yeah, that is a new innovation that could possibly replace the in vitro cell culture assay, but with certain limitations. Right. So what about genomic detection? Current method widely used in genomic detection of dengue is the real-time RT-PCR and serotyping, and the output can be obtained within 4 to 6 hours, including the extraction of the nucleic acid, the amplification process, and the interpretation of the result. Samples, however, need to be taken during the viremic phase. So if it surpasses the viremic phase, you will not be able to detect the dengue virus using this technique. So interpretation is rather qualitative unless a quantitative PCR is requested and performed to quantify the viral copy numbers. But performing quantitative PCR is quite hassle because you have to, if you're doing it manually, you have to build a standard curve and you have to interpret based on the copies number. But having said that, now we have commercially available quantitative PCR kit as well, where they provide the standards in the kit. So it doesn't take that much of time. Well, however, the limitation with real-time RT-PCR is the need for expensive equipment such as real-time PCR thermal cycler. So in resource-limited setting, poor resource countries, where they do not have access to such high sophisticated equipment, this might be a bottleneck. So in order to replace the real-time RT-PCR, a new innovation called isothermal molecular technologies have been recently developed. So what is this isothermal amplification methods? This method do not rely on thermal cycling. Well, in usual PCR setting, you have several range of temperatures where you need to conduct the PCR. Sorry, can you hear me?
Hello.
Hello?
Yes, can.
Okay, so where in the usual PCR setting, you have several range of temperatures that you need to set in the PCR thermocycler in order for you to get the output. But in the isothermal molecular technologies, you only rely on one constant temperature. So the amplification still takes place, but at a constant temperature. So this significantly simplifies the implementation of nucleic acid-based methods in POC diagnostic device. Right, yeah, so here this RT loop mediated isothermal amplification, which is a new innovation in dengue detection. So it is simple and rapid. Within 30 minutes, we are able to obtain the result at a single constant temperature. So it is well suited for on-field applications detectable by naked eye due to colorimetric changes. So as simple as that. And with technology advances, now the lamp has been upgraded, where it has been introduced, such as digital lamp. They have linked it to a smartphone for you to view the results. Some of them have also incorporated biosensor with lamp, and they have also innovated lateral flow based lamp. But the problem with lamp is false positive and non-specific bindings. So these sometimes regarded as the major disadvantages of the lamp method. Another advantage, technology advances in dengue detection, is the micro PCR. So quite interesting, where micro PCR allows the usage of a portable PCR system that can be used to detect dengue. There are also a number of NAAT-based platforms in the pipeline that have potentially been used for diagnosis, but today, only the TrueLab real-time micro PCR system is commercially available. So this system basically uses a chip, okay, that has already incorporated the nucleic acid extraction and the PCR inside this tiny chip, and it has to be inserted into a mini micro PCR, a portable PCR system that can read or interpret the results, also qualitative, positive or negative. So it's quite interesting. We hope to see this micro PCR being used in many laboratory and also hospital settings in future. Right. So antigen detection. So previously we have enzyme immunoassays, ELISAs, and immunohistochemistries, which are quite effective methods for detecting dengue antigens, particularly from frozen paraffin embedded tissue sections using labeled molecular monoclonal antibodies. But their usage is limited in regular laboratory testing due to their laborious method. So instead now, we have tests against intact virus particles and recombinant viral proteins have been established, including commercially accessible NS1 detection POCT. So this POCT is quite widely used in hospital settings nowadays. Okay, what about the antibody detection? So ELISA remains as the was one of the most effective serological diagnostic technique to detect and capture the IgG and IgM particularly. It is quite useful, but the limitation is high cross-reactivity with other arboviruses because basically the flaviviruses, they share similar structure, antibody structure, IgG, IgM. So cross-reactivity is highly evident. So lateral flow assays are more preferred due to early usage and availability of combo version with NS1 detection. We do have some recent advances in this area, such as again the use of biosensors to detect antigen antibody. However, the feasibility of such innovation in the laboratory setting as a POCT is still not evident. So a lot of research or validation still need to be done for these technologies because they have not been commercialized yet. So yeah, moving on to the challenges in dengue detection assays. So what are the challenges that we are seeing? Of course, false positive, false negative. These are the main challenges in dengue detection assay. So a false positive result, basically, it's presented with cross-reactivity. So cross-reactivity with the other circulating flavivirus, as I just mentioned. So particularly the dengue IgM and IgG can give you a false positive result. What about a false negative result? So sometimes NS1 cannot be detected when the level is too low, especially in secondary infection and if the sample has been collected late. So we need the viremic phase to be maintained in order for you to detect the NS1. IgM can also be not detected. It could cause false negative, especially in secondary infection, whereby the IgM, the titer of the IgG is much higher than IgM. So these are among the pitfalls, challenges in dengue detection assay. Another issue is the prediction of disease severity. Until now, I don't think so we have a reliable assay to help the clinician to predict the disease severity, whether this patient going to end up to have severe dengue, and because the prognosis is not good. So is there any test available to predict the disease progression? I don't think so we have any commercially available test kits to do that. So which means we need to look for new biomarkers in this area. So right now, what is best is combining the NS1 detection with IgM, IgG serology in routine diagnosis of dengue infection can help to improve accuracy of the dengue diagnosis. So, so we have come to the end of the presentation. So here are some take-home messages. The future of testing can be important in improving rapid tests that are affordable and sensitive. But there are challenges, especially variation in serotypes, dengue serotypes, and genomic structure is one of the major concerns that need to be considered in the diagnostic development process. So WHO recommends assured criteria, ASSURED, affordable, sensitive, specific, user-friendly, rapid and robust, equipment-free, and deliverable to end users. So any test kits that are able to fulfill the assured criteria will definitely be useful in diagnosis, in diagnosing dengue. So with that, I end my presentation. Thank you.